Okay, welcome to my channel Sudhir Biology. Today we study principle of inheritance and variation, that is chapter number 5, class 12. We come to the next point, chromosomal theory of inheritance. This theory is proposed by Walter Sutton and Theodore Barberi in 1902. They work out the chromosome movement during the meiosis. As you know, gametes are formed by during meiosis. Gametes are formed by the meiosis, right? The behavior of chromosomes are parallel. You can see there are these chromosomes. They are homologous chromosomes. Their behavior are parallel. And behavior of genes and used chromosome movement to explain Mendel's law. Their genes chromosome parallel, genes are also parallel, and that's why they follow the Mendel's law. Sutton united the knowledge of chromosomal segregation with Mendelian principles and called its chromosomal theory of inheritance. They united the knowledge about their chromosome. They follow the behavior pattern of the Mendel's law. That's why he called the chromosomal theory of inheritance. Chromosome and genes are present in pairs in diploid cell. As you know, diploid cell, 2n cell, chromosomes are present in pair. Suppose this is the chromosome, genes are also present in pair. They are also present in pair, and they are segregated during gamete formation during meiosis. They are segregated and they are rejoined during fertilization. During meiosis they are changed into haploid, and during fertilization again they developed into diploid. The chromosomal theory of inheritance claimed that it is the chromosomes that segregate and assort independently. Segregation and assortment किसका हो रहा? Chromosomes are segregate from their pair during the gamete formation by the meiosis. Now experimental verification of chromosomal theory of inheritance by Thomas Hunt Morgan and his colleague. Morgan worked with tiny fruit fly Drosophila melanoglaster. This is the biological name, Drosophila melanoglaster fruit fly. Why Drosophila they select? Because it is suitable for genetic study. It grown on simple synthetic medium in laboratory. It can grow in the laboratory. They complete their life cycle in about 2 weeks. Very short life type. You can say also it have very short life cycle. So we can study many their generation within a year. Number of generation we can study. A single mating couple produce large number of progeny. They produce number of progeny, so we can easily study. Clear differentiation in male and female. There is a sexual dimorphism which is also present. We can identify phenotypically which one is male, which one is female. Fruit fly have many type of heredity variation. They have many type of heredity variation. Linkage and recombination, which is based on the chromosomal theory. Morgan hybridized, that means cross fertilized yellow body with white eye female to brown body and red-eyed male. Morgan cross fertilized between the yellow body and white-eyed female with the brown body and red-eyed male and intercrossed with their F1 progeny. He observed that the two genes did not segregate independently of each other in and F2 generation. The ratio deviated very significantly from 9:3:1 expected when the two genes are independent. Means in previous slide we can study they are independently assorted, separated. But in some cases, in case of Drosophila, there is all genes are not segregated. That's why the result is not found like 9 ratio, 3 ratio, 3 ratio, 1 ratio. Means the result is something different from this. That means we can say there is no all genes are segregated during gamete formation. When two genes in hybrid cross were studied on the same chromosome, the proportion of parental genes combination was much higher than the non-parental type. Morgan attributed this due to the physical association or linkage of two genes and coined the term linkage physical association of genes on a chromosome. Means during division, during gamete formation, the genes are not segregated, they are linked to each other, they are physically associated to each other on the chromosome. Here you can see with the diagram, on the basis of suppose this chromosome contain one gene, and this is the round gene, this is the one gene, this is the round gene, and after crossing over during gamete formation, both genes are exchanged. They are come in this side, in this side, in this side, right? They are linked to each other, they are linked. But in case of in this chromosome, you can see this chromosome contain lined gene, right? And this chromosome contains circular gene. There are two genes are circular. After crossing over, after formation of gamete, you can see one lined gene is here, right? Another lined gene is in this chromosome. One circular gene is here, and one circular gene is here, right? How this is takes place? Because they are present away from distance, right? In between two genes there are different distance is higher than the previous. That's why during crossing over only one gene is segregated. That means all genes are segregated. In this case, genes are segregated during crossing over, but in this case there is no segregation, that is a linkage. But in this case segregation takes place. So recombination, the linkage physical association of genes on the chromosome is called as linkage. Recombination, the generation of non-parental genes. Combination means there are combination of two different genes, means genes coming from two different chromosomes. That's why combination. Morgan found that even when genes were grouped on the same chromosome, some genes were very tightly linked. Those genes are which are very tightly linked, or they are very nearby, they are tightly linked, they perform low combination. As you see, they are not combination, they are similar parental type, they are transferred from one chromosome to another chromosome, right? While other were loosely linked, showed very high recombination. Means those genes which are tightly linked, they show very less recombination, and those genes which are tightly they are loosely linked, or in other words they are far away from each other, then they are having higher rate of recombination. The genes white and yellow were very tightly linked and show 1.3% of recombination, only 1.3%. That's why the ratio 9 ratio, 3 ratio, 3 ratio and 3 ratio does not found in the Morgan's experiment, right? The genes white and miniature wings showed 37.2% recombination, hence loosely linked. In this case, genes are loosely linked. After student used the frequency of recombination between genes pair on the same chromosome as a measure of distance, right? Measure of distance between two genes mapped their position of chromosomes. Recombination based on the distance of two genes on the chromosome. Suppose they are far away, their chances of recombination is higher. If the genes are very close, then there is no recombination. They are very less recombination. Paleotropy. A single gene can exhibit multiple phenotypic expression. Such genes is called as paleotropic. Single gene can perform more than one type of phenotypic expression, then it is called as paleotropic gene. The mechanism of paleotropy in most cases is effect of genes on metabolic pathway which contribute towards different phenotypes. Phenotypically affect. Phenylketonuria, a disease in human, is an example of paleotropy. This is disease which is example of paleotropy. This disease is caused due to mutation. What is mutation? Mutation means sudden heritable change in the generation is called as mutation. So it is mutational change in the genes. The code for the enzyme phenylalanine hydroxylase, which is mutated, resulting it cannot be converted. Phenotypic expression characterized by mental retardation and reduction in hairs. Those persons which are affected by the phenylketonuria, they are mentally retarded and body hairs are very less. Now, next topic, sex determination. Hanking 1891 traced a specific nuclear structure during the spermatogenesis in some insects. 50% sperms received these specific structure, whereas 50% sperms did not receive it. Hankins gave a name to this structure, that is X body. In future, this X body is also called as X chromosome, like this, like X. This is the X body in the nucleus, which is called as X chromosome. But he studied in this experiment, in few gametes the X is present, but in other gametes, in other sperms, X is absent, 0. Now, the sex determination in grasshopper. You can see sex determination in grasshopper is XX and X0 type, like same we study in previous slide. All eggs bears one X chromosome along with the autosomes. Some sperms, that means 50% sperms bear X chromosome and 50% do not X chromosome. Egg fertilized with the X chromosome, resulting female offspring is produced, that is 22 autosome plus XX. Egg fertilized without sperm is without X chromosome, then it is male. You can see this is the female and this is the male, right? This is the female gamete and this is the male gamete. Here you can see XX, XX and X0, X0. So these two offsprings are male and these two offsprings are female. So here you can see male is determine the sex in the grasshopper.